Okay, so last lecture we talked about blood identification, and obviously blood is the #1 body fluid that we see in the crime lab, especially in the biology DNA section. The second most frequent body fluid would be semen, as you can imagine, because it's often left behind in sexually based offenses, and unfortunately we have no shortage of those. So yeah, we process a lot of evidence containing semen. So obviously the presence of semen indicates that there was some type of sexual motivation to the crime. One thing I want to point out though is, especially those of you planning a career in law enforcement, the lack of semen does not always indicate that there's no sexual motivation for that crime, okay? Because remember, we'll get into this in our serial killers unit, which I think is going to be the unit, maybe we'll do it next, okay? Kind of, jazz up the class for midterm. But these are not normal people, and oftentimes they can not function normally sexually, so they may not be able to complete the act. So even though it's a sexually motivated crime, you may not have any semen left on the body or on evidence near the body. So, keep that in mind. But obviously, if you identify semen, that indicates that some type of sexual gratification was a motive in the crime. By the way, this opening photograph, this is a scene from Ferris Bueller's Day Off, if you've ever seen that movie. That was when I used to talk about saliva too. But we're stopping with blood and semen identification. Okay, so the types of evidence, once again, we've talked about how anything can be physical evidence. So obviously anything can have semen deposited on it. But if you were to look at the most frequently seen types of evidence that have semen on them, a sexual assault evidence collection kit, which is collected from a victim who goes to the emergency room and says, I was sexually assaulted, and that contains swabs of the victim's orifices. That's a very common evidence type to examine. But inner garments such as underwear. So if ejaculation happens, in the vagina or in the rectum, if the victim is alive and then they stand up and walk around, very common to see drainage of that semen, onto the crotch of the underwear. So we look at that a lot too. But anytime, if the victim's able to tell you where ejaculation occurred, it can be on outer garments, it can be on bedding, tampons, okay? What job can you work where you actually get excited about, getting a used tampon in as evidence? But in terms of a sexual assault case, that is a really excellent piece of evidence because a tampon is absorbent material. So not only will it absorb the semen from the offender, but then you're also going to have cells from the victim. And so it's pretty easy to be able to tie the victim and the assailant together. So that can be a really useful piece of evidence. Have I ever, come in contact with cases where a biologist examined a diaper for the presence of semen? Unfortunately, yes. Things that we don't like to think about, but people who sexually assault children, there's no real age limit. So yes, unfortunately, that is not an uncommon or incredibly uncommon type of evidence, unfortunately. Condoms are actually, here's another example. What kind of job can you work where you get excited about a used condom and you having to examine it? But once again, that is a perfect type of evidence because you're going to have the semen from the offender ejaculated into the condom, and then the epithelial cells or cells from the victim on the outside of the condom. And so once again, it's a perfect way to link the offender to the victim. And what I love is that a lot of offenders, they believe that if they use a condom, it will destroy the DNA. Not true, okay? So even if an offender is using a spermicidal condom or some type of spermicide, all spermicide does is it prevents the sperm from swimming. And so it can't make that journey, up through the cervix and the uterus and the fallopian tube, and it's to prevent pregnancy, but it does nothing to destroy the sperm head, which is where the DNA is. But, you know, we're glad that the bad guys don't know that. So keep leaving those used condoms right at the scene, because it's a perfect way to link you to the victim. As I mentioned before, remember, these are not normal people. And so a lot of times if, you know, a serial offender, which, you know, normally they are male, and I'm not trying to stereotype males in this, but obviously semen comes from males only. And the higher percentage of sexually based offenses are committed by males. So remember, these are not males that can usually function sexually in a normal way, and so it's not uncommon to have inanimate objects used to penetrate the victim. And that may be for, you know, post-mortem posing of the victim. It may be to punish them. The offender might be angry because he's not able to complete the act. You know, there's a lot of different psychological reasons for that. Let me mention at this point too that unfortunately I learned pretty early on in my career that women are no less evil than men. And especially women who, you know, abuse or kill their own children, but they tend to use different methods. So that's why, you know, most serial killers, which tend to be, at least the ones that we hear about in the news, which are sexual sadists and sexual predators, tend to be males. And then you really don't hear that often about female serial killers. Of course, Eileen Wuornos would be an exception to that, and we'll talk about her. You know, they're no less evil, okay? They just tend to kill their own children, or, you know, do types of crimes where they're partnered with a male. So yeah, women, I'm not cutting women a break here at all, okay? I've seen it all, unfortunately. So back in the old days, okay, and the old days would fit into when I first started out doing forensic serology when I was down at Southeast Missouri State. So this is going to sound so gross, but it's basically you felt the stain and then you kind of looked at it and smelled it. And oh yeah, you know, it kind of has a dry, crusty, whitish, yellowish appearance. Yeah, it could be semen. Let's do some further testing. Thank you, Jesus. Those days are gone. Okay, now we can use other potential presumptive tests to identify semen. So one of the ways it can be used is by shining an ultraviolet light on an item of evidence. And we refer to this as the ALS, or the alternate light source. Okay, so semen fluoresces under UV light. Okay, however, so will urine. And, you know, oftentimes if you're looking at maybe the, you know, the crotch of the underwear, you are going to see fluorescence, and it may be due to urine, but that's why you do confirmatory tests, okay, or take things further. But semen definitely fluoresces under UV light, okay? So let me show you an example of a piece of evidence where this would not be readily apparent, and then the ALS is really useful. Okay, so here we have a belt, and if you look at the belt, this is a photograph just taken under normal light. Okay, so we have this darker brown stain here, and if I were to look at that, if anything, I would say maybe that's a blood stain. You know, let's do a KM test on that and see what we come up with. But lo and behold, you shine the ALS on it, and look at how it fluoresces. Okay, so that is actually a potential semen stain. Not that there might not be blood mixed in with it, but the fact that it's fluorescing indicates that, yeah, we need to do some further testing to see if this, you know, seminal material is present. Okay, so first of all, let's talk about what semen is. Okay, so semen is the fluid that a male ejaculates, and semen and sperm are not the same thing. And we actually have confirmatory tests for both sperm and semen. Okay, so you can have semen that doesn't have sperm in it. Okay, a man who has had a vasectomy still can ejaculate, but there's just not going to be any sperm present in that semen. Okay, so semen is technically defined as the fluid in which sperm travel, and the chemical composition of semen is it contains sugars, which are basically there for the sperm to use for metabolism, so it can be able to swim on its incredibly long journey, if you think of it in terms of a molecular level. There are also secretions from the prostate gland, and semen has a slightly basic pH. The reason for that is the vaginal environment actually has a slightly acidic pH, and so when semen hits the vaginal environment, it neutralizes the acid, and that is a much more hospitable environment for sperm to begin their journey. Okay, so that's kind of the reason for that. So we have forensic laboratory tests that then they are confirmatory tests to look for both sperm and semen. But we also have a presumptive preliminary or screening test for semen as well. Okay, and here's just a fun fact. Once again, you don't have to memorize these numbers for an exam, but a typical male ejaculate is between 2 to 6 milliliters and has up to about 150 million sperm per mil. Okay, and when pregnancy happens, you just hope that the good sperm, with the good traits makes it there, but it's kind of a crapshoot. Whichever sperm reaches the egg first and is able to penetrate it, ding, 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 that's your winner. And then the egg actually becomes impenetrable to all other sperm. Okay, so the anatomy of a typical sperm cell is as follows. So up here, this is the sperm head, okay? And that is where all of Dad's DNA is. At the very tip, you have a cluster of enzymes called the acrosome. And the acrosome enzymes are there to help the sperm head actually drill through the cell wall, or the very sturdy cell membrane of the egg, which is actually pretty tough to get through. So yeah, it's kind of a way to help that sperm head drill so it can make its way into the egg. Okay, so the DNA that we're going to talk about in the next set of lectures from Dad only comes from the sperm head. Okay, now this is going to be important too when we talk about mitochondrial DNA. So if you can flash back to your basic biology class, you may have heard about mitochondria, right? Oh, sausage shaped, the powerhouse of the cell. Okay, and it's where the energy in the form of a molecule called ATP is produced. And ATP is needed by the sperm cell so that it can swim all the way to the egg. And so this middle piece here contains tons of mitochondria. And mitochondria have their own little teeny tiny piece of DNA. Okay, so that's Dad's DNA too. However, what happens once the sperm penetrates the egg, the sperm is decapitated, and so the only thing that enters the egg is the sperm head with Dad's DNA, and then the middle piece and the tail fall away, okay? So there is no mitochondrial DNA from Dad that makes it into the egg cell. It's only from the sperm head, okay? Or what we refer to as nuclear DNA. You'll get into that in future lectures. Okay, the other thing about DNA analysis is we don't really care if that sperm is normal or abnormal. Okay, if I worked in a fertility clinic where I'm trying to help couples have babies and, figure out what the problem is, like this guy, the sperm head is way too big, it's probably too heavy, can't swim very far, this guy's too little, these guys are going to be swimming, in a circle, here you have two tails. I mean, these are all abnormal sperm that could affect fertility, but at the DNA lab, we don't care about that, okay? We just want the DNA component so that we can help identify who the offender is. So yeah, sperm structure, as long as we have a sperm head, yay, that's good enough for DNA. So the preliminary or presumptive or screening test for semen is called SAP, which stands for seminal acid phosphatase. Now, phosphatases are enzymes that basically cleave off phosphate groups, and you have them all over your body, okay? They're in lots of different body fluids. For example, there's vaginal acid phosphatase, VAP. We used to use that as a preliminary test. We don't use that anymore. But the reason that SAP can be used as a preliminary test is that seminal acid phosphatase is up to 1,000 times more concentrated in semen than in other body fluids. Okay, so it's not a confirmatory test, but if, you know, we get a positive presumptive test, it can tell the forensic scientist that, hey, you know, there's a good indication that semen might be present in this sample, and you should go ahead and do a confirmatory test. So the test for seminal acid phosphatase pretty much works exactly the same way as the KM test. It's a color test. It is, remember, a presumptive test. The only difference really, you know, besides different reagents being used, but it's, you know, dropping 2 reagents and then you see what color it turns. The only difference is that in seminal acid phosphatase, it will turn purple if it's positive, as opposed to the KM test for blood turns hot pink, okay, or a pink color. So here's an example of different semen samples, and they basically took semen and then they diluted it. And so this is the most concentrated, and you can see it turns this purple lavender color. And then as you dilute the semen, it gets lighter and lighter. The SAP test is not as sensitive as the KM test. You know, that's one of the drawbacks to it. But it's still, I mean, you know, it's pretty sensitive. And, you know, it can at least give you an indication that semen is present. Okay, so that's the presumptive test or the preliminary test. Now, there are 2 different confirmatory tests for semen. Okay, so one, we're gonna look for sperm cells and actually look by, you know, putting a sample from whatever type of evidence, maybe it's a vaginal swab, you know, from a rape kit, and we're gonna take a little bit of that and we're gonna put it on a microscope slide, and we're actually going to look for the presence of sperm. So if you identify sperm, you've automatically identified semen. Okay, you cannot have okay, let me rephrase this. You cannot have sperm without semen, but you can have semen without sperm. Okay, that's where the vasectomized males fit into. So this first test is called the KPIC, and I'm not gonna give you the big long, you know, wordy definition of what that is, but it's basically a microscopic staining technique to look for the presence of sperm. And again, this is where the forensic scientist ranks it from negative, meaning that they don't see any sperm or sperm heads present, because remember, the heads are the important part. That's where we're gonna get our DNA. 4+ would mean that they see many sperm heads. So yeah, it's a little subjective, but a 4+ KPIC result tells you that, you know, you can use less of the sample to try to do DNA. That's a really good indication that, yep, we have a lot of sperm heads. There's a really good chance we're gonna be able to get a DNA profile from that. Okay, so the stain that's used on the KPIC is called the Christmas tree stain. And, you know, we're not trying to be light about the subject. We know that we're looking at sexual assault evidence. It's simply called that because the 2 different dyes that are used are green and red. Okay, so for us in the DNA section especially, if we see a lot of red staining, which stains the sperm heads, that's really good news. That means that, yep, we definitely have semen identified because we've identified sperm, and there's a good chance that we're going to be able to get a DNA profile from that. And then the green stain just picks up the sperm tails. Okay, so let me show you an example of what this looks like. Okay, so this would be the view looking under a microscope of a positive KPIC, and I would say that this is like a 3+, 4+. So notice here where it stains red, these are all of the sperm heads. And you literally need 1 sperm head to be able to get a full DNA profile. That's exactly what happened in the beauty shop rapist case, right? They had a microscopic slide and there was a single sperm head and they cut it out of there and, you know, were able to get a full male DNA profile, which matched Dennis Magruder. So yeah, you just need 1 sperm head to give you a full DNA profile. In this case, we have a whole bunch of them. So to me, that would indicate, yeah, I don't have to use that whole swab. I can maybe use half of it and then you preserve the other half in case the defense wants to do their own testing and send it off to another lab. So that's where the KPIC, I think, can be really useful. Okay, the second confirmatory test is called P30. And P30 is another name for prostate-specific antigen. This is a protein that is found only in semen. And you may have heard of it called the PSA test. Okay, so fellas, when you reach about the age of 40, your doctor, probably on a yearly basis, is going to ask you to take a blood test for the presence of PSA or P30. The reason for that is that P30 or PSA should only be produced by the prostate, and it should only be found in semen. And if you can detect it in the blood, that is an indication that for some reason it's leaking into the blood, and that can be an indication that there may be prostate cancer present. So that's why the PSA antigen is really important to test for, usually when a male hits about 40 years of age, you know, just as a check for potential prostate cancer. In terms of semen identification, it's super sensitive. You can literally take semen and dilute it 1,000,000 times and still be able to identify it. Okay, and the beauty of this test is we don't need to identify sperm. Okay, we can pick up offenders that potentially have had a vasectomy, maybe they have a low sperm count, maybe for whatever reason they didn't fully ejaculate, but there's some pre-ejaculatory fluid that was left on the victim or at the scene. This test is going to pick up those guys because they don't have sperm, and this test doesn't require sperm to be present to detect semen. So it's really a very powerful test, and it's really easy to do. So it uses a pre-prepared test that we call an abacard, and it literally works just like a pregnancy test, okay? So a typical pregnancy test, the woman holds the test into her urine stream, and then you let it sit for a little bit, and you know, if you see 2 lines, you're pregnant. If you see 1 line, you're not pregnant. This works the exact same way, except we're looking for the P30 antigen. Okay, so for example, here I'm showing you 3 different tests, and it looks exactly like a pregnancy test. Okay, so let's say we have a vaginal swab that we think has semen on it. So we might take a cutting of that, soak it in some sterile water, and then take that sterile water and inoculate it where it says S. S stands for sample. So this is the sample we're testing, okay? Then what's going to happen is the sample and potentially the P30 is going to wick up through the test, and in, you know, I think it's about maybe an hour, we can come back and we can read the test. Okay, notice that we have 2 lines up here. C stands for control. This is our positive control. So if we don't see a C line, that means that the test didn't work and it's invalid, and we have to repeat that test. Okay, T stands for test, and this is the line that will show you whether P30 is present or not. So this is what a positive test looks like. It worked because you have a line where C is, that means the control worked, and you have a line where there's a T, that means that P30 was identified. Here's a valid negative test where this, you know, the control worked just fine, but there's no line. That means that there is not semen detected because nothing showed up where the test should have showed up. This is an example of something that if you ran this and the C line doesn't show up, that means, okay, you know, I got a bad test, the control didn't work, it's invalid, we need to go back and repeat it. Don't assume that this is a negative because the control didn't work, so we can't make any assumption about that sample. We need to go back and do it over. Okay, but obviously if you get a test like this, and you can test a whole bunch of samples at the same time, that indicates, yep, semen is identified, not indicated, because it is a confirmatory test. And then, you know, normally that sample will go on to DNA to see if we can develop the profile of that male contributor who left this semen behind. And what you'll learn about in the DNA section is we also don't need sperm cells to get DNA from semen samples, okay? Every time a male ejaculates, There's going to be what we call epithelial cells from the lining of the urethra that are carried out into the ejaculate, and you can get DNA from those cells. Or, same thing with a urine sample. That's going to have those cells in it too, because the urine is traveling out through the urethra. So because DNA testing has become so sensitive, we can pick up a lot more offenders, okay? It used to be using the old technique, you had to do a K-pick and there had to be sperm identified, or basically that guy got a free pass. Okay, not so anymore, and thankfully because of the Abacard and looking for P30. Okay, so that concludes the semen lecture, and now we're going to move on to see how we can do DNA on these samples and try to develop, who potentially could have contributed to these different body fluid samples.